yeah, so after all of these fantastic viruses in eukaryotes, I will actually take you back to the microbial world. But kind of a similar story, or at least some similarities. And specifically, there will be a lot of virus discovery. And I targeted a group that so far has been mostly overlooked, I would say, in our exploration, at least of the virosphere. And these are these inoviruses. So what are these inoviruses? You may know them as filamentous bacteriophages, which basically is just a name for their weird virions particle, which has this filamentous shape here. A few dozen of nanometers long, a few nanometers wide. They have relatively small genomes, single-strand DNA genome, 5 to 20 kb, mostly circular. And one other very interesting feature of these viruses is that they have this persistent infection cycle. And if you were here for my Roux talk yesterday evening, you already heard about persistent viruses in plants. This one is slightly different from what she described. These phages infect bacteria and can stay and reside for a prolonged period of time in the bacteria at least without killing the host. But they can still produce virions because they use the virion production machinery, which is an extrusion mechanism which is not directly killing the host. So basically, they get the best of both worlds. They are transmitted vertically because they are inherited from bacteria to bacteria, and they can transmit horizontally. Now, you may also know them because they have been used for decades in biotechnological application. If you ever heard about phage display is all based on inoviruses or filamentous phages. There have been other more recent innovations, pretty cool stuff like a nanoscale battery, which is also all based on these filamentous capsids. And there is a lot of talks about using this type of phages for non-traditional applications like a vaccine carrier in human. At the same time, beyond biotechnological applications, they have also major effects on certain bacteria, let's call them bacteria of interest. And the main one that you probably heard about is Vibrio cholera. So you all know this bug, you all know the disease. What you may not know is that the cholera toxin is actually encoded by one of these inoviruses, which is itself integrated into the Vibrio genome. So basically, the Vibrio cholera is really only really nasty when it's itself infected by a virus encoding this toxin. And that's kind of an extreme case, but we have multiple other cases where we have seen these specific phages having impact on the host phenotype, which just like repercutes on the pathogenicity and toxicity of the bacteria. And that has been seen in plants and in humans with species like Neisseria. So at this point, we're like, OK, we have a group of viruses. We know they are great for biotechnological applications, and we know they have massive impacts. We must know a lot about them. And that's when the fun comes in, because actually we don't. Right now, these are represented by a single viral family, 39 genomes isolated from only 10 bacteria genera. Basically, we have inoviruses for beta and gamma proteobacteria here, one from Actinobacteria, a few from Deinococcus, actually 2, and a few from Spiroplasma. And that's it across the whole bacteria tree of life. Now, if you were here yesterday, or even if you kind of followed the recent literature, you were like, oh, yeah, but no, we don't do isolation anymore. We do metagenomics a lot. Well, if you go to the largest database of genomes from viruses assembled from metagenomes, which is IMGVR, hosted at JGI, you have the grand total of 0 inoviruses. So there is something weird here. Either we are not sampling them well, which could be the case because they have a weird capsid, or we just don't detect them well. And that's kind of my talk here really today is, as you guessed, we actually sampled them, but we couldn't detect them until recently. And since we did find a way to detect them, it kind of completely changed at least my perspective on these viruses, and especially my perspective on how they probably interact with their microbial host. OK, quick technical part. How do we discover these new inoviruses? Typically, when you want to find a new member of a group of viruses, you start from a marker gene. And typically, we start from the capsid, because the capsid is clearly coming from a virus. Problem is here, their capsids, and if you even just compare the isolates, they have absolutely no sequence similarity with each other. There is absolutely no conservation in these inoviridae capsids as soon as you come across a broader, like more than 10 genomes. There is one single gene which is really conserved, like slightly conserved. It's like 40, 50% amino acid identity, so it's OK in these viruses. And it's unfortunately, it's an ATPase. So ATPase are so promiscuous that you will find tons of genes in every microbial genome that kind of look like an inovirus ATPase, but will not be an inovirus. So how do we solve this? Well, these ATPase are still kind of a good starting point, but then what you have to look at is a genome context around. So basically, you can't find sequence similarity, but you can still find defining features. For example, we know inoviridae have a lot of short genes. If you compare the median length of genes in an inoviridae genome, which is here, compared to random fragments of the same size in a bacterial or archaeal genome, you have this clear difference. So that's one defining feature. We actually found a few other ones. Inoviridae will have more uncharacterized genes. Inoviridae will usually have one gene, which is relatively short, 100 amino acid, with a single transmembrane domain covering the whole sequence. And that's pretty typical from their capsid protein. So no sequence conservation, no primary sequence conservation, but a 3D structure or 2D structure relatively conserved and that we can detect even from the primary sequence. And if we want an Inoviridae, we want all of this, or at least we want a combination of these features in a region which will be 5 to 20 kb, because that's kind of the expected genome size. So when I say this like this, it feels like kind of hand waving. Oh, yeah, it looks a bit like this, it looks a bit like that, which is basically what we need to do, training a machine learning algorithm and then having some kind of a systematic way of classifying these viruses. And the best way we found is using a random forest classifier where you can get sensitivity of more than 90%, and especially you can get a very, very low false discovery rate. So basically, what we call potentially new Inoviridae based on these features is most likely a new Inoviridae. OK. Some biology now. Started with the genomes. We started from 65,000 genomes. We got almost 8,000 hits for ATPase. And our tool tells us 4,500 were true inoviruses, including 1,500 complete. No, we had these genomes. That's all microbial genomes. Obviously, we have metagenome assemblies. We have tons of metagenome assemblies. So we also mined these ones. A lot more hits. Assemblies are not as good as for genomes. So the numbers are not as good, but basically we had another set of almost 5,000 inoviruses. So again, we started from 39 isolates, and now we have 10,000 of these sequences. Technically, this could all be the same sequence, but that's relatively easy to check. The way you define an innovirus species, according to ICTV, is 95% ANI over the whole genome, so 95% identical nucleotide. And when we did that, we ended up with more than almost 6,000 species. And if you do an accumulation curve, you can see that we are very, very far from saturation. So basically, we keep finding new species of inoviruses as we go through and look into more data sets. And what is amazing is that's the red is the accumulation curve in the microbial genomes here. But then when we add the metagenome sequences, it's kind of having another bump. So basically, what we find in the metagenomes is very different from what we find in the currently sequenced microbial genomes. So a lot to discover here. OK, so that was the part where we started from 39 genomes, and now we have 6,000 species. What do we do next? The first thing I was very interested into is just come back to this host range. So remember, right now we have isolate genomes for inoviruses infecting gamma and beta proteobacteria mainly, a few spiroplasma, one actinobacteria, and 2 isolates from Deinococcus thermus. And that's the new picture. Every group in red has now at least one genome of inoviruses associated with it. And that's actually the number of genomes here, so number of viral genomes associated with each group. And that's a log scale. So it's kind of a bit misleading, but basically we still have most of our detection, actually 90%, in these gamma and beta proteobacteria, which makes sense. They have been massively sequenced in terms of microbial genomes, so that's quite what we expect. Now, you might have noticed a few things very cool beyond the fact that we have a lot of red. His group here is CPR, this candidate phyla radiation. We have inoviruses for 7 different clades of CPR. And we have detection in archaea. Actually, I should have, yeah, come back. So here, these are actually archaea, and we have inovirus-like viruses, well, ino-like viruses, let's call them, in one Enigma-Archaeota genome, which is a metagenome assembled genome, so that's kind of the end of the story. We can't really do anything with this one. But we also have them in 3 Urearchaeota, which are isolates. And obviously, we thought, hey, we should cultivate this thing and try to induce a prophage, because that will be the perfect back-to-the-lab experiment. So we enlisted the help of Rep Dali from the Wrighton lab, and she was able to grow one of these isolates, which is a metanolobus isolate. This is what the cell looked like. And this is really going on and still very early. But basically, what we are very excited about is like when we grow this cell at high density and when we stress them with mitomycin C, we start to see this kind of clumps forming, which are not incompatible with what clumps of filaments will be. So I'm not claiming these are inoviruses yet, but we're on the good path, at least in my opinion. OK, good on time. Wrapping up here, very quick story. I don't have to present this, but basically when we do a taxonomic analysis of based on gene contents, remember we had one family of Inoviridae. We have now what we think is a full viral order divided into 6 different families and nearly 300 subfamilies. So that's kind of the real extent. And remember, the accumulation curve is not saturating yet, so this will probably grow. Now, what I was very curious about is we know these viruses tend to reside in the host cell for very long, so they have to have some very interesting interaction with the host. And specifically, I was curious about how they did that with hosts encoding CRISPR-Cas system, which is supposed to be an immune system that will just get rid of this type of viruses. So the first question was, are these targeted by CRISPR-Cas system? And indeed they are. When we match these genomes to the collection of spacers that we have from known microbial genomes, we get more of 1,000 of these inovirus genomes with protospacers, so matched to spacers. And these spacers came from more than 1,000 host genomes. And they actually came from all types of CRISPR systems, type 1, type 2, and type 3. So we definitely know they are targeted. Next question is, do we see self-targeting, which means, do we have a bacterial genome where we have an inovirus prophage which is targeted by spacers from the same genome? So for that, we just look into our host genomes with a spacer matching an inovirus. About half of them had also an inovirus prophage. So at this point, you have a genome, you have one or more inovirus prophages, and you have spacers matching inoviruses. However, we got only 19 self-targeting. So said otherwise, usually if you have a bacterial genome, you can have prophage of inoviruses, you can have spacers matching inoviruses, but these spacers usually match an inovirus found in another member of the same species or another member of the same genus. At this point, I don't know if this is because cell targeting, which is supposed to be, or at least thought to be very lethal for the bacteria, is just very, very strongly selected against, or if there is any mechanism by which these inoviruses can evade sometimes spacer acquisition. Now, of course, as always in biology, I was most interested by the exception here. What happens with these 19, and how do they live in a bacteria which is supposed to target them constantly? So we know there are a few anti-CRISPR mechanisms, a few proteins have been described. We looked for this in our data sets. Basically, across almost our 80,000 sequences, we had 2 low similarities, so nothing that's been convincing of a potential anti-CRISPR. However, we can still look at the content of the genome, and we had a few examples that were very interesting, and that's one example. This is an inovirus prophage here. This is a CRISPR array from the same genome. You have all the caste genes, and you have the spacer here, and you have 3 hits from these spacers to this prophage, and the hits are 100% identical, and we have protospacer adjacent motif, so everything should work. In this prophage, you have all the virus gene here, and then you have a bunch of uncharacterized gene here, including one with an HTH domain, which is a DNA binding domain, which is very typically found in anti-CRISPR mechanism. So arguably, this is all speculative. This is sequence-based. But I would say if you want to find new anti-CRISPR mechanism, you should look into viruses that have very prolonged residence time in the host cell. First on your list should be these inoviruses. OK, just a few words and take-home message for conclusion. We largely underestimate these inovirus diversities. There is no question about this. And they are clearly a full viral order with multiple families and not just one single family. They associate with all types of bacteria. I didn't show it, but we find them in all types of biomes, including hot springs, hydrothermal vents. They are basically everywhere. And I also didn't show it, but they are also infecting bacteria that are infected by other viruses like codovirals. And we don't really know anything about virus-to-virus interaction in this host cell, but that could be interesting. They have, in my mind, a very unexpected functional richness. Again, they're a relatively small genome, 5 to 20 kb, but we have tons of different genes in these genomes. I showed you the example of anti-CRISPR. Again, we have viruses from this extreme environment. And remember, this has these filamentous capsids, which are all based on one single protein, which means we probably have here systems to do flexible filaments in very extreme environments. And there was a lot of homologous replacement and a lot of evolutionary link to plasmids. And finally, what we provide here with this type of work is really kind of a first step. We did all this. We found all these new viruses. That's cool. But there's also a new framework for anyone else to try to look into these genomes for whatever their protein of interest or their virus interaction of interest or anything. And with that, I just have to thank everyone from our groups at JGI for the CRISPR program, the Royton Lab at OSU, and Mark Rupovich from Institute Pasteur to just help me with all this. Thank you very much.